What I'd like to do is just give a short 5-10 minute overview, a little bit of background on Alzheimer's disease, give you a little bit of background on some of the topics that we're going to start to discuss tonight, and a little bit of more information on the ways that we're using stem cells to understand how Alzheimer's disease works. I'm sure most of you are aware of a lot of these statistics, but just to really reiterate, this is a devastating disease that's affecting many people now and many more people coming in the future. So by the time people reach their mid-70s, 80s, up to 1 in 3 people will die with Alzheimer's and dementia. That leaves the other 1 or 2 people that are caregivers, and I think that's an equally important take-home message, is that the task that we're going to be asked to do for our loved ones with Alzheimer's is almost as challenging as the disease itself, and so we have a great need of being able to address this disease. It's going to be a very costly disease as the population ages. More and more funds are going to be needed to address this disease and treat it. Alzheimer's has been diagnosed by a couple of different features. One is degeneration of different nerve cells in the brain, particularly in areas that are responsible for memory and other aspects of really higher cognitive function. But if you look at, a pathologist looks at the brain, they find two really sort of telltale signs of the disease, and those are these protein aggregates that are initially described amyloid plaques. These brown spots that you see here are aggregates of this beta amyloid that, if it's formed in the wrong form, it will stick together and basically aggregate, and you find these as a sort of a diagnostic marker post-mortem. So these are images that are taken of brains of patients that have passed away with Alzheimer's. Another feature is these tau tangles, tangles of protein that form inside the neurons that eventually lead to death of those neurons. We've had some success in over the past few years in using different imaging techniques to look at these markers in living patients, and this has really allowed clinical trials to progress and help us understand a bit more about the progression of the disease. So there are lots of developments in different ways of imaging both tau and amyloid in the brain, and watching this disease develop. So we have some tools to start to address clinical trials. But there's still this huge unmet need. So as most of you are aware, almost all of the clinical trials that have been approached in Alzheimer's disease, that we have an extremely high failure rate. Only about 5 symptomatic treatments have been approved. And these often don't really, they're not disease changing. They can treat some symptoms and delay progression for a very short amount of time, but we currently have no drugs that actually stop or cause a change in progression of the disease. A lot of something we'll probably talk about tonight is just announced today, another failure of a drug that was another failure of a drug where we're trying to use antibodies to pull amyloid out of the brain in a family where the people that have genetic mutations are guaranteed to get this. This so far has not resulted, this strategy has not resulted in a way of actually stopping progression of the disease such that it preserves cognitive function and aspects of the disease that were sort of primary endpoints. So we'll talk a bit more about that tonight. But what I wanted to do is give a little bit of background on an approach that has been developed over over the past 30, 50 years of understanding genetics and how genetics is leading to ways of understanding Alzheimer's. It led to being able to address some of the initial clinical trials in Alzheimer's, try to target amyloid and tau, and that's still a big component of therapeutic approaches, but it's also genetics is also leading us to understand some potentially new ways of targeting the disease, and that'll be a topic of discussion tonight. So there's essentially two different types of general types of Alzheimer's. There's rare genetic mutations in one of three different genes that if you have that mutation, you're guaranteed to get Alzheimer's. So these are rare cases. There are families around the world that carry these mutations, and usually they start to get Alzheimer's in their 30s or 40s. The vast majority of people that get Alzheimer's usually start to get Alzheimer's later in life, in 60s, 70s, 80s, and older, and that the genetic component of that is very complex, and we're starting to understand a lot about how that how different genes might increase our risk for developing Alzheimer's. But one of the things we're struggling to understand, and where a lot of the research focus is going into, is how those different genes and genetics actually function to contribute to the development of Alzheimer's disease. And some of you have probably seen there's a few mutations that in Alzheimer's, the genes that are actually protective. And so, for instance, this Christchurch mutation that was described in one of these families that has the mutation that guarantees that they get Alzheimer's. This mutation in a gene called ApoE that we'll talk about in a second allowed her to actually survive much longer without any signs of the disease. So it gives you a potential handle, and we'll talk more about this, a handle on ways, therapeutic ways of potentially intervening. So we're just asking, can we use genetics to try to drive some therapeutic development, and we'll talk more about that as the night goes on. Like I said, in the vast majority of people that get Alzheimer's disease, there's a number of growing different disease genes that might increase our risk for getting Alzheimer's disease. I think this is from last year, and I think we're up to about 25 to 30 different genes that have been identified. I think one of the main ones that we're going to discuss tonight is this ApoE gene. This is a gene that causes probably one of the most gives you probably the highest risk of getting Alzheimer's disease. And this gene sort of comes in three different flavors. So most people have this gene called, a version of this gene called ApoE3. And if you remember your genetics, you have two copies of these. So most people, the vast majority of people have two copies of ApoE3, but there are fewer people that have different numbers of this ApoE4 allele, and other people that have different numbers of this ApoE2 allele. And what's interesting about this is that the mutations that have been discovered in this that associate with Alzheimer's actually affect the protein. They affect its shape and how it might interact with other proteins and other molecules. So it gives us sort of an interesting handle on thinking about how to therapeutically intervene there. And what is sort of, I think, hopefully setting us up for an interesting discussion around how to use ApoE is this, that in most of us have this 3-3 version, and over time, and about when we start reaching 80, 85, about half the people who have this mutation will start to show signs of Alzheimer's. People that have the two copies of this 4 allele have a much worse case, so they start getting it sooner, and it can be much more severe. But if you notice this ApoE2, if you have two copies here, you're largely protected from Alzheimer's. There are very few people that have ApoE2 alleles actually get Alzheimer's, and if you add just one of those two versions to the 4 version, you can improve things and actually move this line closer to what most of us get. So we'll talk more about how this is working and how clinical trials are being designed to take advantage of this fact. But what about understanding all of these other risk alleles? So we can We've identified many of these risk genes. They're in different types of cells. They might be in different people and participate in different mechanisms of the way that the disease develops. And so the way that we've been looking at this here at NICES is to use stem cells as a way of modeling this in the laboratory. So we can create avatars of ourselves by creating stem cell lines from people with and without the disease. We have access to their genetics, so we can manipulate their genes and these different risk factors and understand how those manipulations can impact the way that the biology carries out in the cells that are actually affected by disease. So this gives us the opportunity to develop new therapeutic approaches. So if we can take blood or skin from anybody here in the room, we can create a stem cell line. These stem cells are very much, they look like embryonic stem cells. They can make any cell type in the body. We can use our understanding of development to push those cells to develop into, those stem cells to develop into the different types of brain cells, in this case the brain cells that are involved in Alzheimer's disease. And in doing so, we can basically replay the disease over and over again in the dish and try to understand what's the first thing that goes wrong in those cells that are actually succumbing to disease. We can make a lot of the different cell types and main cell types in the brain, oligodendrocytes that ensheathe the neurons, microglia, the immune cells in the brain, and the target of many of these genetic risk factors. But you can find these genetic risk factors in almost every single one of these cell types. And so being able to replicate this in the laboratory and then even start to mix these together into structures that we call organoids, or little micro tissues that allow the cells to interact much in the same way that they do in the brain. And this gives us the opportunity to tease apart what happens as the cells talk to each other and understand how the genetics of those patients that gave those cells, how that affects the way that these cells communicate with each other, and find handles that we can use to better develop therapies. So we've shown that we can actually be able to see a lot of the features of Alzheimer's in these different cases, and we can use things like CRISPR editing and genetic tools to correct some of these mutations and manipulate and understand how these are working. So we'll talk a bit more about this as the night goes on. But there's still a lot of challenges here, but also opportunities for new therapeutic approaches. And there's challenges that this may be not one disease, but maybe many diseases, different subtypes of disease, each driven by a different underlying genetic component that we would like to be able to understand, and using things like stem cells to tease apart these complex disease mechanisms. Traditionally, we've used mice to study these different genes, and the stem cells allow us to complement that with actually looking at human cells that give us the opportunity to understand things that might not happen the same way in mice that they do in humans, and we'll talk a bit more about that. So I hope this sort of sets up some of the different topics that we're going to talk about, and we have lots of opportunity to dive into the details. So I'll hand it over to Max to get us started with discussion.
All right, so in terms of FDA-approved standard interventions, as Scott mentioned, these are only symptomatic treatments. They do not change the progression of the disease. They help symptoms in some people for a little while, but by a year and a half in, they always wear off. So they're really unsatisfactory even as symptomatic treatments. So for, I guess, about a century ago, Alzheimer described the accumulation of these protein clumps in the brains of a woman, a 55-year-old woman with a disease that now bears his name. In the 80s, we began to understand or discover genes that were linked to Alzheimer's disease by looking at DNA sequencing of patients with the disease. That led us to focus on or converge on these amyloid plaques. You saw pictures from Scott and Max mentioned them in the therapies. So the first clues that we got about the genetics and the pathology pointed to amyloid plaques, and they're still a defining feature of the disease. This is a normal protein that is misfolded and clumped, and as it builds up, kills nerve cells, perhaps because of or together with causing inflammation in the brain. Some of the first genes that were discovered were amyloid genes, or the first Alzheimer genes that were discovered were genes that encoded amyloid. And that then really sort of concretized the field, and we began focusing on reducing amyloid buildup as the be-all to end-all for Alzheimer's disease. And for the past 30 years, we've, as Max mentioned, we have come at that both with small molecules, drugs, and with antibodies. And we've been able to totally eliminate amyloid buildup in the brains of people with Alzheimer's disease. And despite the fact that their brain scans now look pristine because the amyloid is gone, removing the amyloid has had nothing at all to do with the progression of the disease.
The question, and I'll throw this out to the whole panel then, is it we're just not intervening soon enough? By the time we work to get rid of the amyloid, the damage has been done, and we need to do something much sooner in the process here? Or is it that amyloid is simply guilt by association? They're just innocent, maybe not totally innocent bystanders, but they're not really involved with the disease process at all. Ron, you want to take a shot at that? We're going to get back to you on the genetics here in a minute. But
well, the concepts in terms of the pathogenesis of Alzheimer's is as I mentioned, amyloid has been the initial focus. But then later in the disease, you begin to develop these tangles called tau tangles, and that's thought to be an important target as well. But there's also inflammation related to the disease, inflammation in the brain, and that also is thought to be a target. And so it's very, very complex, and some of the approaches that we and others are taking is basically to ignore all that and deal with the genetics.
so let's get into the genetics. Scott, you mentioned before there are different versions of this ApoE allele or gene, 2, 3, and 4. If you got 4, you're out of luck. If you got 3, most of us are kind of somewhere in the middle, and if you've got 2, you don't have to worry about it. Tell me how these things seem to work.
So ApoE.
Yeah, I'm working at it. I think it's on. You just have to aim it right at your mouth.
Okay.
There you go.
There we go. So ApoE, like I said, it comes in basically three different flavors, and the mutations that give rise to these 3 different flavors change the shape of the protein, and they do so in such a way that it might alter the way that those proteins bind or interact with other molecules. In particular, the role of ApoEs, one of the roles may be to shuttle things like cholesterol and lipids around and among different cells in the brain. Or, ApoEs found in the brain, but it's also produced in the liver and is really, important for cholesterol metabolism and the way that those.
Which is when we first heard about ApoE4
yeah,
was really relating to heart disease, right?
Yeah, exactly. And so, there's been a lot of work looking at genetics, looking at populations of people, thousands of people that have different copies of these genes. And what it appears is that there's a dose-dependent response of the number of copies of the ApoE4 that gives you worse and worse sort of outcomes with respect to Alzheimer's. But also the flip side is going the other direction towards ApoE2 that seems to be more protective, and then different combinations can pull things one way or the other.
So this harkens back to the ApoE2 story, and it was discovered in '93. There were a couple of families, sort of like this Christchurch story. But in this case, these were also families where there were mutations that would guarantee Alzheimer's, but there were escapees. There were people who should have the disease who did not. And when their genome was scrutinized, it turned out that they had ApoE2. So there arose the concept that if you have the right flavor of ApoE2, that can protect you against Alzheimer's, even if you have the world's worst gene that causes Alzheimer's disease.
And Ron, what this then opens up is ways to try to figure out therapeutic angles here, ways to get at it, because Do we know how these genes cause Alzheimer's or lead to Alzheimer's? And then how do we intervene?
So
is the question.
there's literally hundreds of articles in terms of why these genes either protect or cause Alzheimer's. So the approach that we've taken is to ignore all of that and to go basically to use the epidemiology that we've now heard a good deal about. E4 is bad, increased risk, E3 is average, and E2 is decreased risk. So think about it, if we could convert the brain from an E4 to an E2 E4, we could do the same as if you inherited E2 from your parents, or even give more E2 and convert you more toward an E2. So the question is then, how do you do that?
How do we do it?
And so that's what the gene therapist studies. And the way we use it is we take the normal gene, in this case the E2 gene, and we put it in a virus called an adeno-associated virus. This is a virus that doesn't cause human disease. We gut it from all the viral genes, and we use the virus like a Trojan horse. We administer it to the fluid surrounding the brain, the cerebral spinal fluid, and the concept is that it will bathe the brain in this good gene, the E2 gene, and its product, the protein, and therefore take away the risk. We've demonstrated that, at least in mice, that really works. We know we can do it safely in non-human primates, and we've now started a clinical study, and we have already dosed 2 patients with it. It's an outpatient procedure, and of course it'll take years to determine whether it works or not, but that's the concept.
I mean, to think that you're born with 2 copies of ApoE4, normally that would increase your risk 10, 12-fold. Almost no one with 2 copies of E4 escapes Alzheimer's. And the ApoE4 genes are present in at least half of patients with Alzheimer's. So it's probably the most potent, most common, arguably the most important Alzheimer gene that will ever be discovered. So I think that after 30 years of failure aiming at amyloid, now we let the genetics of the disease, where we know how the genes behave in humans, let the genetics lead the way, and we follow behind. Rather than trying to understand every step, we go straight to these silver bullets that seem to be able to prevent the disease.
We've seen that it seems to work with mice, but mice ain't people. Why not? I mean, how different are mice here in these circumstances, and can we rely on what we've seen in mice to be reproducible in humans? Do you think?
Well, with a 40-year plus history of doing clinical research, one thing that I've certainly learned is humans are not just big mice. And you can use mice to learn and do very sophisticated, interesting, and important studies that lead you to suggest that it works. But eventually you have to do the human studies. And so you have to show first that it's safe, and if it seems to be safe, then move to humans and then try to work it out.
Michelle, we mentioned organoids before and putting these things. What use are organoids in studying this kind of an approach? Refresh our memory as to what Scott mentioned about organoids, first of all.
Want to answer this one?
So organoids are basically assemblies of human cells that we've created from stem cells, and we've been able to make different cell types, different neuron cell types. So one of the cell types that we can make are these neurons called basal forebrain cholinergic neurons. They're some of the first to potentially die in the disease. We can also make, and what may be important for understanding some of the neuroinflammation, we can make things like microglia. So these are the basically the innate immune cells of the brain. They're responsible for basically gobbling up proteins, dead, dying cells. During development, they help sort of hone and create the different circuitry that develops in the brain. They have lots of different roles, and they are potentially one way in which humans might differ from mice. There's some similarities and some differences in microglia in the brain, and so being able to have both to look at is quite important here.
If I could really emphasize how the iPSC field has helped us, I mean,
I mean,
prior
you
to
you
the
can't say iPSC fields without telling us what that is.
the stem cell field.
Okay, there we go.
I mean, prior to this, you know, we used mice or rats, and then we went to non-human primates, and then we went to the humans, and there was nothing that substituted for the live human. And although the stem cells and the organoids do not substitute, they really have already been invaluable in identifying some of the differences, for example, between mouse and man that Scott alluded to. In fact, for specific, you know, there was a slide that Scott showed that showed, you know, the 20 or 25 risk factors for Alzheimer's disease, and it's already been shown by multiple stem cell groups that several of the proteins encoded by those risk factor genes act much differently in humans than in mice and interact, and I think what's even more important, using the stem cells and the organoids, we're learning how some of these risk factors interact with each other, including ApoE. And although, you know, I totally agree with going with the epidemiology and ignoring the science at times, I think what these other experiments allow us to do, and I think it's already happening, is find more and more targets that can be hopefully pharmacologically druggable without actually going after the amyloid or the tau.
I think what I would like people to take home is that aside from, you know, the different news articles that keep advertising failure, I think there's a lot of different new ideas of ways of thinking about how to treat this disease. Even just simply understanding that it may not be one disease and it may be many diseases, that we need to understand, you know, different ways of thinking about how to treat those. And certainly I've placed, you know, my bet on using stem cells to understand that as a potential approach. So I think there's a lot of optimism, and I think these ideas are developing very quickly. Simply understanding that it's not one disease and maybe many is a huge step in the path to actually finding specific therapeutics for each one of those.
Great. Michelle?
I think what I would emphasize is that the scientists and National Institutes of Health has really been in the vanguard in terms of promoting this, is that we are looking, as I mentioned before, for other approaches to Alzheimer's disease that do not have to attack amyloid or tau, since those have been such failures. So, for example, nerve cells communicate by connections called synapses. And there's a lot of work now directed at determining how inflammation affects the synapse, and can we correct or at least improve the connections between nerve cells without actually attacking the amyloid or tau. And I think experimental models are really key for looking for this, and also working with big pharma to try and repurpose drugs that can be used to go after some of these pathways, which is a lot less expensive than developing entirely novel drugs.
Aside from all the things that Sam mentioned in terms of health and in general health, and doing the New York Times crossword puzzle to keep your brain in shape, I think the most important thing is, without research, there's no cure, and support biomedical research. Not only the foundation here, at a federal level, the National Institutes of Health, and so on. And unless there's research, we're not going to cure these diseases.
So first, if I only told my patients one thing, and could be sure they would do it, the one thing I would recommend is physical exercise. It's really the best way to keep your brain healthy these days, despite the fact that it's a very low-tech piece of recommendation. We've mentioned these multiple genes. So there are 30 genes that are associated with Alzheimer's disease now. A third look like protein folding genes, maybe related to how amyloid and tau form. But another third are due to fat and lipoprotein metabolism, and the last third are associated with the immune system and inflammation. We don't know which of those molecules comes in what sequence, and whether we want to go with a drug that turns them up or down, or when to do that. We're really sort of, the more we know, the less we know. And I think that the idea of letting the epidemiology, the genetic epidemiology, now lead the science, this is an outstanding time to do that, as Ron is pioneering.
Thank you, Sam. So I think I've been doing a lot of Alzheimer's panels for a number of years, and this is, I think, really one of the first times that I've come away with some hope that we really do have some avenues that I think are going to be very productive here. Also, though, please remember that, as Ron mentioned, if you don't take part in clinical trials and you don't help us out with research, nothing gets done. So that's another important thing to remember, is we have to get involved, we have to volunteer. We also have to be the squeaky wheel for research funding. That's how research gets funded in this country. If you make a lot of noise, somehow the money shows up for that funding. So if you're interested in research funding for Alzheimer's, make a lot of noise about it. I want to thank our panelists for something that has really been, I think, very interesting and very hopeful. Thank you all. This has really been a terrific evening, and I believe we still have some refreshments left over there in the back. So thank you very much for being here.